Good morning, everyone. I think we should begin. People will trickle in as the conference proceeds. So I want to welcome you. My name is Ruth Ottman. I am Deputy Director of the center that is sponsoring this event. It's a center with a long name, the Center for Research on the Ethical, Legal, and Social Implications of Psychiatric, Neurologic, and Behavioral Genetics. In a moment, our center director, Dr. Paul Applebaum, will tell you a little bit more about the center. But I just want to welcome you and say that, we're really looking forward to this conference today. So let me introduce Paul, who'll tell you more about the center. He is the Elizabeth K. Dollard Professor of Psychiatry, Medicine, and Law here at Columbia. He's past president of the American Psychiatric Association and the American Academy of Psychiatry and the Law. His research today focuses on ethical and legal issues related to the use of genetic and genomic data. He's served on multiple committees and received numerous awards. He's a member of the National Academy of Medicine, and he's also a prolific writer and commentator. So we're really pleased to have him.
Thank you, Ruth. Good morning, everybody. Thank you for joining us. My task here this morning, before I turn it back to Ruth, is just to tell you a little bit about our center, which is sponsoring this conference. Ruth read you the name, so I won't take up the time it takes to do that again. But we're funded by the National Human Genome Research Institute as one of 6 centers of excellence in ELSI research, so-called SEERs, around the country. The purpose of which was to create research centers with the resources and expertise to design and implement multifaceted and multidisciplinary investigations of particularly complex, persistent, or rapidly emerging ELSI issues. And for those of you who aren't familiar with the acronym, that's ethical, legal, and social implications of genetics and genomics. We've been in existence now. We're just about at the end of our 7th year of funding, initially as a developing center and for the last 4 years as a full-fledged center. And in addition to the research that we conduct, we have a large number of other activities. We have postdoctoral fellowship. We have 2 fellows, ELSI fellows, at any point in time. We do a monthly ELSI seminar here at the medical center. If any of you are interested in being on the email list for that seminar and aren't yet, please let me know. Just give me your card or jot down your email address, and we'll be sure to add you to the list. You're at the annual conference, so you know all about that. We do a series of topical workshops over the course of the year. We have a website, which I'm going to show you in a moment. We do short courses on methodology for faculty and trainees here at the medical center. We support pilot studies. We're very involved in the university's precision medicine initiative. I'm one of the co-chairs of the Precision Medicine and Society program, along with Alondra Nelson, who's the Dean of Social Sciences on the Morningside campus. And we were asked by NHGRI to plan with them this year's ELSI Congress, which will be taking place in June 5th through 7th, on the campus of the Jackson Laboratories on the UConn Medical School campus in Farmington, Connecticut. The program looks outstanding, I must say, and if any of you are interested in more information, feel free to stop me at the break, and I'd be happy to send that to you. This is our website, which I would invite you to visit. The URL is braingenethics, one word, columbia.edu. And you'll find all kinds of information about the other things that we do. And one of the things we do, and the last thing I want to tell you about, is our Brain Gen Ethics update. This is a monthly summary and compilation of literature related to the ethics of advances in the genetics of psychiatric and neurologic disorders and behavioral traits. And we also try to keep our readers updated on the latest genetic advances themselves to which the ethical issues apply. If you would like to receive this free on a monthly basis, you can give me your email address and we'll add you to the list, or you can go to our website and sign up there. So with that, let me thank you again for joining us. I'm enormously grateful to our speakers. We've got a wonderful group of people and looking forward to their presentation. So Ruth, let me give it back to you.
Thank you. I just want to give you a little bit of background about why we decided to have this conference on this topic. Neurodegenerative disorders pose an amazingly challenging public health problem. And this is only going to increase as time goes on. And despite a lot of excellent research, the development of treatments has really not been successful to date, with few exceptions. And one of the main problems is that the biological processes that lead to these disorders begin long before symptoms become apparent. So the most promising approach to doing research for new treatment development is really to target participants who are at high risk for the disorders but don't yet have any symptoms. And with advances in genetic research, of course, we have the ability with genetic testing to identify people who are at high risk. So there are a lot of studies going on now focused on a range of different neurodegenerative disorders that do just that. They target participants who are at genetic risk. This raises a number of ethical challenges, and that's really why we wanted to have the conference on our topic, which is ethical tangles in neurodegenerative research. Now, I should say that this whole range of issues was brought to my attention and to all of our attention by Karen Marder. I would not have really realized the importance of this without her help. And Karen, thank you so much. You've been really a partner in the development of this whole conference. So I really, really appreciate that. And we should all recognize your contribution. Okay, now a few housekeeping things. First, I think you've all realized by now that there are no food or beverages allowed in this room. That's probably the most important housekeeping item. I will be fired if you bring any food in here, so don't do that. And refreshments will be available at the break in a room down the hall. Maybe most of you have already found this room, so just as you leave this area, head down the hall, and there's a room on the right where we have coffee and snacks set up. We've allowed time for discussion after each of the talks, and Matt Leibowitz and Shauna Benston, who are fellows in our center, are in the back there. You want to raise your hands? They will help you have a microphone for the discussion sections. We have 10 minutes for discussion after each talk. And we really encourage that. Of course, we want you to participate. And after the conference, we really would appreciate it if you fill out the evaluation form. We take these very seriously, and we really hope to have a high participation rate, speaking as an epidemiologist here. So one other thing, if you need the restrooms, they are also down the hall. They'll be on the left-hand side, and they're well marked by signs. Okay, so with that, I believe we can begin. Our first speaker is that we're really very happy to welcome Dr. Irish Olson, who comes to us from Georgetown University, where he is professor of neurology, pharmacology, and human science, and also director of the program for regulatory science and medicine. He has played an enormously important role in research on a range of neurodegenerative disorders. He founded the Parkinson's Study Group and the Huntington Study Group, and these are international academic consortia whose main focus is research on neurodegenerative and neurogenetic disorders and also treatment development in those disorders. He's held leadership positions in more than 35 clinical studies of Huntington disease and other disorders. He's received numerous awards and is a member of the National Academy of Medicine. So we're very fortunate to have him today. This it will advance the slides and also as a pointer, right. Or you can use the keyboard.
Which one?
I think the down key.
Okay.
Okay.
Go.
And that microphone.
Well, thank you, Ruth and Paul, for inviting me. Actually, thank you for inviting me back, because I was at one of your early conferences, I think just when the center was starting, and really was very impressed by, I thought you had been around for 10, 20 years already by the time you were doing that one, but I really think this is such an important center, not just to recognize the importance of ethical, legal, social implications of research related to genetics and genomics, but also to disseminate information. And I think also you probably mentioned, or I'll mention that the slides and the presentations will be available publicly for people to use. So I think that's really an important part, and data sharing is a real big piece of this too. So this is the title I have, but I'm going to be talking about the Pharos Study, which I actually talked about 5 or 6 years ago, but I'll give you a one-decade look at this relatively unusual study, certainly unusual when it started out. But if you just indulge me for a second, I just want to mention, had a hard 2 weeks, lost 3 real important people in my professional life. Mike Keneally, who many of you know is a really important genetic epidemiologist, Peter Como, a neuropsychologist, one of my fellows, and then recently Bud Rowland. And Bud Rowland had a real big impact on me. One, he was editor in neurology and taught me how to write a scientific paper. But he also, and this was before it was fashionable, recognized the importance of randomized controlled trials. Although he wasn't a clinical trialist himself, he really questioned people in the early days about their contentions that such and such a therapy was really effective. This is actually from his obituary, which is in the New York Times, but I guess I knew this, but I thought it was really interesting because if you knew Bud, he's a tough guy. And his family name is Rosenthal, but it was changed to Rowland so he can get into an Ivy League school at the time when Jews were really not admitted. And he went to Yale, where he was an undergraduate, got his MD degrees, went into the Navy, came out. It was interesting when he was at, I didn't even know this existed, there was an association of interns and medical students at the time. Of course, we take this kind of for granted now. And he was the head of that. But that led to a big problem because when he went to NIH shortly thereafterwards in 1953, he was interrogated by the FBI in the McCarthy era about his connection in leading this kind of left-wing organization, and he refused to answer questions, and he was fired from NIH at the time. I don't know if people knew that, but this is really a remarkable person. He really had a fierce devotion to justice, but also to beneficence and autonomy, so I thought it's fitting to mention him here at a conference on ethics. So just a few disclosures. I am on a oversight committee for one of the studies that will be talked about today, the Colombian study that Jason will be talking about. I'm also on an oversight committee for NeuroNext. I'm an executive director of a small biotechnology company that works in the domain of neurodegenerative diseases, and also recently co-founder of a new company called Gray Matter Technologies. All these things I mentioned actually have no bearing, but I just wanted you to recognize, because it wasn't, part of the disclosures. I won't talk about any of this, and it shouldn't influence my presentation, but you be the judge. So here's what I'm going to talk about today: the Pharos Study, prospective Huntington at-risk observation study, and these are the topics I'm going to discuss. I have a lot of detail in some of the slides, which I'm going to seemingly gloss over, but they will be available afterwards, so in case one wanted to really dig into the data. Actually, the weeds here are very important, but we don't have enough time to, get into that. So first of all, the background for the study. So what is Huntington's disease? Most in the room know, but it's a neurodegenerative disorder of genetic etiology that affects about 30,000 individuals in the U.S. who have manifest HD, and about 5 times that number who are at risk for developing HD, this autosomal dominant disorder. Actually, Mike Keneally was the one who taught me how to figure out the 5 times the manifest, just because of the age of onset of the disorder. The etiology is genetic. It manifests itself through neurodegeneration, which I'm not going to talk about hardly at all today. But I will talk about the important phenotype that results from this, because the study that we did is really a study of phenotype, of clinical phenotype, and I think that this is really very important. So here's the gene etiology. Nancy Wexler led the group in 1983 that identified the linkage to the gene, and then eventually 10 years later, a lot of things happen after 10 years in this business, but about 10 years later the actual gene was identified, and I was pleased to be a part of that. But I think the important piece to recognize is that as an autosomal dominant disorder, one allele at this locus, which is on the short arm of chromosome 4 at what's now called the Huntington gene, one allele has a normal number of CAG, cytosine, adenine, guanine, repeats at this locus, a normal number being up to about 20, 30, 32, 33, and you can see the distribution here in green. But on the other allele, there is an expanded CAG repeat, and that's what you see in the red. And this can be expanded anywhere from, we use the number 37, we could talk about how critical that is, all the way up to 120, 130. And of course, the important recognition here is that the extent of expansion is inversely correlated with the age of onset. So the higher the expansion, the earlier the age of onset. In fact, the children, almost even infants that we've seen with Huntington's disease, have expansions in that range. But to recognize the typical adult onset has this part of the red curve with a lot of variance in terms of age at onset there. So you just can't look typically at an age of onset. You can't look at a CAG repeat expansion and know the age at onset. So, as I mentioned, it's inversely related. It's felt that this expansion probably accounts for 50 to 75% of the variance in terms of age at onset, the other part being, quote, environmental or epigenetic factors. In fact, it even could be greater than that, although this is an area I'm not going to spend too much time on. One thing to recognize as we started this study is that CAG as a precise number. It's looked at in the clinic. In fact, we've done a study sending DNA to molecular geneticists, and they're usually within 1 or 2 repeats of one another in terms of doing it. So it's a reliable, precise measure. But we're talking about age at onset. How precise is that measure? And that's one of the things we're interested in looking at, because to the extent that we could postpone the age at onset in individuals who carry the mutant gene, that would be tantamount to almost disease-modifying therapy. So most of the work. Oops, that's not good. Yeah, I think I hit the wrong part of it. Yeah. So most of the focus in experimental therapeutics has been when people manifest illness. And we've been doing a little bit better over the years in terms of detecting and diagnosing Huntington's disease. And in fact, if you look at this cartoon, before 1990, 1995, this is when we were diagnosing Huntington's disease. As we really understood phenotype better, we were getting a little bit better at it, but it was also obvious that there's a lot of stuff happening before we would diagnose Huntington's disease, and not just in the motor domain, but also in terms of cognitive, behavioral, what we call the precursors of Huntington's disease. So the question we asked is, to what extent are these precursors predictive of actually developing HD, and to what extent could they be used in a clinical trial aimed at postponing the onset of HD? So the other thing that was really evident to us at the time is, as the gene was recognized and discovered and clinical testing was underway, that only about, at least in North America, only about 10% of individuals who were at risk for HD chose to undergo pre-symptomatic DNA testing, that's what we call it now, and the other 90% not. And interestingly, that's not really changed that much, at least in North America, over the past several decades. And although in the people who we see in research, we see a lot of people who undergo pre-symptomatic testing. There's a little bit of a sampling bias there. But in general, in the people in the at-risk community, there is either a conscious decision, I don't want to be tested, or more likely than not, they may not even know they're at risk for HD, or they deny they're at risk for HD, so they don't get tested. So what we thought is, why don't we do an observational study where we could compare those who don't carry the gene, the non-expanded, to those who carry the gene, and be able to do this study by learning actually their gene status, but never disclosing it to the research participant, to the investigator, to anybody, to the family, to anybody in terms of any kind of individual data, only pooled collective data, as I'll show you. So that was really the basic part of the study, and that we were going to follow you about every 9 months for, we said originally 3 to 5 years, and it ended up to 10 years as we extended the study to get more information, of course, with the consent of the research participants. So the purpose is to identify these early precursors in their specificity for whether they have the expanded repeat or not, and we plan to enroll 1,000 consenting research participants at immediate risk for having inherited the gene, but have chosen that they don't want to learn about their HD gene status. And as we think about Alzheimer's disease and now Parkinson's disease and other disorders, this kind of replays itself in terms of, do I want to know what effect is it going to have on me, and how I change my mind over time about knowledge of gene carrier status. And then we were going to follow these individuals. And these are people, only adults we were looking at from the age of 26 to 55. And we wanted to see the proportion of subjects who, over this period of time, as I said initially 3 to 5 years, develop signs of HD, And the major consent stipulation in the study, and this was important, is that research participants will not be informed of their expanded repeat status, and if they or they will not be told if they develop clinical signs of HD unless they asked, and if they asked, then we would arrange, or they could go to their clinician who took care of them, or we would find somebody to take care of them and discuss this outside of the context of the study. So that's how we handled this. We kind of isolated it. And by the way, I remember in the early days we were writing this grant. If not for Karen Martyr, who kept taking me through the scenarios about what if and what would happen, I don't think we would have ever really had the sufficient foresight to do this. So Karen's kept me honest here for about 20 years in this study now. So how do we decide HD? Now there are other things, but there's a lot of literature suggesting that cognitive, intellectual impairments antedate the motor impairments, and I think that's true, but the real question is the specificity. How much is this specifically linked to the HD gene? So we decided that almost arbitrarily, these are mostly neurologists, but we decided arbitrarily that we would look at motor features of HD, namely the involuntary movements, especially the choreic type of movements, but there's also dystonia, choreoathetosis, myoclonus, a whole bunch of other movements. But that we would look at these movements and then develop this confidence scale up to a 4, where there was 99% confidence by the investigator that he or she actually had a extrapyramidal movement disorder that was related to HD. And that was a 4, and then we also arbitrarily decided ahead of time, we pre-specified the first time they get a 4 is equivalent to the research diagnosis of HD. So that's the background for the study. How did we do? Well, this was our early publication in 2006. We did pretty well, and we actually enrolled over a period of 3 years, 1,001 individuals. That last person who came in, we just couldn't cut off the 1,000 in time. And what you see here, interestingly, is the difference of almost a 2-to-1 ratio of women to men in the study. And we've seen this pretty much over and over again in our studies, and also, well, particularly in Huntington's disease, but we see it in other studies. And one, without going into details, women will participate as research participants in observational studies where there's no immediate gain, much more so than men will. It's almost 2-to-1. And why is this? Well, I think it is partly they're more altruistic. They have the greatest reproductive responsibilities too, and things like this. So we see this time and time again. If you go into a clinical trial in Huntington's disease, it's almost 1-to-1 ratio. There's equal number of men to women. And this is kind of an interesting phenomenon. The only thing I will say is it didn't have any difference on the study, because in Pharos across the board, we didn't see any differences in our outcome comparing men to women. And here's the baseline characteristics of the study. Really no differences between the men and the women. Average age in the study was 41, which is pretty close to the average age of onset of HD. And as I said, these are the categories. And at baseline of these 1,001 individuals, the overwhelming majority who had no signs, or what we call very soft signs, really represented what this cohort was. There were a few people who had grade 2 motor abnormalities, just a few grade 3. We actually had 10 people out of this 1,001 who, at the time, although they were enrolled in cohort, were diagnosed by these criteria as having HD, and we just continue to follow them in the trial, but we've censored them from the data going forward. But we did pretty well. 99% was a clean cohort. So what are the how did we do over these 10 years? What are the relevant events? And is it feasible to maintain this confidentiality? So I really like to look at feasibility in clinical trials. Are almost as important as the therapeutic outcome. You know, how can you do this? So at the end of the day, when we analyze the data of these 1,001, let's say 1,000 individuals, we found out that 64% of them had normal or non-expanded CAGs, and 36% had the expansion. Now, this is important. This is actually what one would expect in this type of population. We're pretty close to what one expects if you just looked at all comers who are at risk for Huntington's disease. And what we found during the course of the study is that there were a number of events that we had pre-specified to look at in the study that turned out to be different between those who had the expansion and those who did not. And these differences are highlighted here in the yellow, but the group that had, and unbeknownst to them about their expansion, at least the vast majority of them, had more exacerbations of depression, new onset depression, suicide risk assessments prompted new mental health evaluations, psychiatric hospitalizations, and suicide attempts. These turn out to be relatively small numbers for the 1,001 people at risk. The other things in terms of how long they stayed in the study and medical events were really no different between the groups. So at least in terms of reportable events, we considered this a safe study. Now, we also gave research participants these confidentiality surveys to fill out, I think at every visit where we sent them in the mail. We thought we were very avant-garde in the time we actually were using different coding systems to do this. I mean, nowadays you kind of laugh in terms of what we're dealing with, but at least in terms of 1999, this is what we were facing. So we looked at subjects who, during the course of the study, on their own, chose to disclose that they were at risk for HD, because we said we wouldn't even say to anybody that you're participating in the study. By the way, this was being developed at the same time working with our congressperson in Rochester, New York, Louise Slaughter, who was the author of the Genetic Information Non-Discrimination Act. So we really had an important role in terms of here was an experiment going on about that. So we looked at people who chose to, on their own, disclose that they were at risk for HD, and there really didn't turn out to be much of a difference between whether they actually carried the expansion or not, nor disclosure by others. These are usually family members or friends. So at least in terms of our commitment to respect this confidentiality, we did well from our end, at least in terms of what we're reporting here, and there was no difference between the expansions. So if you look at feasibility and safety in the study, we think it's very feasible to be able to do observational studies, and I will contend also clinical trials in this population, and that the confidentiality can be maintained. In fact, one of the things maybe we could talk about during the discussion is we have really husbanded our database and only either analyzed it ourselves or under strict scrutiny, or to colleagues such as Karen Martyr and her collaborators. But on the other hand, we want to be able to make this database available, but we have this consent restraint. Well, we could talk about that later, how we might handle that. So, and we think we also learned a lot about this at-risk population, but one is don't be complacent, continue to monitor, especially mental health. I'll show you in a bit why. And especially suicidal ideation, and there is an attrition of research participants after a while, but we have really good retention up to at least 5 years in the study, some, as I said, up to 10 years in the study. So suicidality, important to look at. This is a paper that came out at the end of last year. Primary author Karen Anderson. We wanted to examine those factors associated with decision to be tested and examine the impact on the testing. And I just want to take you to this population where we looked at these types of events, behavioral events in the population, and comparing them. And I'm just going to skip ahead right here just to the point. What we found out there was really more suicidal ideation, as I guess one would predict, amongst the expanded group. And the features, the clinical features associated with that were hopelessness and anxiety as we measured this independently. In fact, they increased the risk from anywhere from 25% to more than a doubling. And although we didn't have any serious outcomes there, this is a population in general, the at-risk population, that is subject to suicidal ideation, that needs to be monitored for it, and also the signals for this are often expressions of hopelessness and anxiety as we were able to measure it. So they're higher in those reporting it, and one needs to be mindful of this going forward. Now, in our population, remember when we started, everybody came into the study saying, I don't want to be tested, but we also recognized early on that people might change their mind, because changing minds in the setting of uncertainty is very common. So we actually, of this 1,000 individuals, it turned out that 10% of the entire population of this 1,000, or 101, I think, decided during the course of the study to go out independent of the study and to get tested for the HD gene. And this is how we looked at this. But what I want to bring your attention to is it turned out that the people who decided to be tested disproportionately were individuals who had the expanded repeat. Of course, they didn't know this at the time, or at least they weren't obviously conscious about this or articulating as such, but it turned out that these were the individuals who turned out to get tested. And it also turned out that this group differed from the non-expanded repeat of those individuals to be tested, these 101 individuals, by not only their CAG repeats that was more common in that group, as I mentioned, but they also had this higher behavioral score, behavioral types of problems. Now, this is really interesting. We, in this group, unbeknownst to their gene status, we looked at a measure of affect or depression, in this case, the Beck Depression Score. Prior to them being tested, we did this in the entire cohort, but this is the group we pulled up. Prior to them being tested, the time they were tested, the visit after being tested, and the next visit after being tested. And what we found is people then who were tested and informed they did not carry the expansion had a low level of depression coming in there and maintained that low level. People who found out, who had the expanded repeat, who found out after testing that they indeed had it, had a higher level of depression coming in there. And not only that, after the testing, the level of depression intensified. Now, this is a little bit contrary to what you read about in some of these anecdotal studies coming out that people undergo testing and they're just fine afterwards. Well, their period of follow-up is usually about 6 months. And I think there's a little bit of a cautionary tale that this is something that continues and should be monitored closely. So after testing is an important time to monitor this. Environmental factors. I'm going to not delve into this in great detail, and I'm going to refer you to also Karen Martyr, who along with Carly Tanner, did a lot of this work. But in this cohort, we collected so much phenotypic data probably too much or not enough, depending on what your point of view is. But we collected data on diet. We collected, of course, data on weight. We collected data on leisure activities, physical activities, and the like. And so what did we find? Well, I'm going to skip this slide. So first of all, we looked at lifestyle exposures, and just want to tell you, we didn't really find much at all. It doesn't matter if we were looking at tobacco or alcohol use, you know, the typical thing, head injuries, types of medications that people take. We did find this little difference in birth control pill use, but I can't make anything out of it. I don't think it amounts to anything. I think it's just by luck. One thing that we did find, and we have no explanation for this, and therefore we're not really pursuing this too much, is that there is a higher level of caffeinated soda use amongst individuals who had the expansion and phenoconverted. And so there's a risk of 2 to 3 times as much in this population. Again, it just might be by chance. Dietary intake, I think, turns out to be very important because we also were able to show prospectively over time that there was a difference between the CAG expanded group and the non-CAG expanded group in terms of their body mass index, and also in terms, and this is Karen Marder's work published in 2009, and also in terms of their caloric intake and body mass intake. So it doesn't matter if one knows their status. This is something that goes on and begins fairly early in the course of the evolution of HD. So this is the conclusion, and probably the diet piece is the most important, that increased caloric intake may be necessary to maintain that, and probably related to the increased energy expenditure, whether it's intrinsic metabolic or it's due to subtle motor activity, is unknown. So I'll skip that. Okay, so let me get to, at least this is my take, that environmental factors may influence motor phenoconversion, especially vis-a-vis diet and the maintenance of body mass, but these factors are really dwarfed by the genetic power of this disease. This is, whether it accounts for 60% of the variants or 70% of the variants or 80% of the variants, it's a very powerful effect. Of course, the fact that we didn't see much in this population, even though there are 1,000 individuals, may also reflect that we just didn't have sufficient sample size to detect that. So what happened to this whole motor phenoconversion that I mentioned in the beginning? What did we find out, and how might this be applied to clinical trials? Well, about a year and a half ago, we published this summary of the associations in the Pharos trial, and again, this is the same scale that we used in terms of the diagnostic competence of motor activities. And here is the cumulative phenoconversion rate over this period of almost 10 years amongst this population. So 1,000 individuals into the population. And you could see we had 2 people rating them independently. One is a site investigator who was aware of all the clinical data, the demographic data. They would meet with him like any clinician. And then we had a motor rater, an independent rater, who just came in and did the motor part of the examination. And you could see there was a higher phenoconversion rate amongst those who were able to get other clinical information in the study. And is that necessary in future studies? I highly doubt it. So this is the comparison of individuals in the study who didn't have the expanded repeat versus those who hadn't the expanded repeat. No difference in terms of age, education, male, parental descent, but obviously motor scores were much worse, so were behavioral scores, so was the cognitive measure. In this case, we used single digit index, but we did a lot of cognitive testing. And over this period of time, there was a slight loss of function amongst this group, but it was so slight that in this very early group, we couldn't really detect many changes. I just want to, almost everything on the motor examination showed an association with motor phenoconversion. I mean, that's expected, but also almost all the cognitive tests we did also showed an association there. And so Korea was the most powerful predictor of phenoconversion, but also we saw changes in cognition over time. Now here's kind of like the money slide of what happens over time. If you look at the total motor score in this population, you can see at the beginning of the study, the group that had the expansion actually was a little bit more motor impaired than the group that did not. And the group that did not have the expansion didn't change over time. But the group that had the expansion, and these are points of phenoconversion over time, you can see it was kind of a steady increase over time, almost a straight line. And this turned out to be the most powerful relationship there was. Interestingly, behavioral, there was a difference between the groups at baseline, but this didn't change too much over time. If there was a behavioral problems, they kind of persisted over time. They didn't necessarily get worse over this period of observation. The non-expanded, obviously, they looked like they even got a little bit better over the period of time. Single digit, a little bit difference at baseline, but these curves diverged over time, so cognitive impairment was very prominent in the course of this. And also functional capacity, although overall it changed, this is on, it changed very modestly on this scale, which is probably not sufficiently sensitive for this. So, how do we conclude in terms of if we wanted to do a clinical trial in an at-risk population like in Pharos, is this a feasible thing to do? How many people would it take to detect an effect on motor phenoconversion, as we showed in the study? Well, here's the bad news. And the bad news is it would take hundreds to thousands, depending on what we assume to be the effect size. Yeah, if you assume there's a 50% effect size, which is pretty big, it doesn't look too bad. But most experimental therapies are not that powerful. It turns out, at least in terms of our analysis, that the motor domain is actually more powerful in terms of a outcome measure in a study than the cognitive domain, at least measured by single-digit modalities. But they're fairly close, and total functional capacity, of course, is an insensitive measure to look at this. So, so where do we stand? Remember, if you did a study in the at-risk population in a clinical trial, at least 2/3 of that population would be non-expanded individuals. And, you know, what do you do in that type of situation? Well, we've learned a lot from other studies, not just Pharos, but Predict HD. I'm not going to spend time on that. Janet Williams will be talking a little bit about that later. Another observational study cohort, 2 observational studies cohort in North America, registry in Europe. A very elegant study on biomarkers, Track HD, and an ongoing study supported by CHDI called Enroll HD. So we don't have any lack of observational studies in HD, and they've been very beneficial and informative. I'll just mention that this PREDICT study went on at the same time as Pharos, or pretty well at the same time, and included individuals primarily who were aware of their gene status, in fact, almost exclusively. So these were that 10% of individuals who decided to be tested to learn they carried the expansion. There were controls in the study of people who were non-expanded, but people were aware of their gene status in the study, and this group had done also a remarkable job. But what I want to point out to you, as I said, we didn't look too much at the brain in Pharos. We looked at the genetics and we looked at phenotype. This group spent a lot of time looking at not just the brain but other markers, and I think we're able to show over a comparable period of time that there were other clinical markers or biomarkers that might be really very useful, particularly striatal volume. They spent a lot of time standardizing MRI studies. So I just want to leave you with a question we could discuss later, because we're going to hear about the Alzheimer's studies that I think in many respects have been more innovative, at least in terms of clinical trials, than we've done. But even to this day, about 90% of at-risk individuals choose not to undergo genetic testing. Some at-risk individuals do still want to participate in research but don't want to learn about their genetic status. To some people this seems puzzling, but at least from my point of view, just because of the respect for autonomy, this is the way it is, and it's the way I hope it'll continue to be. And by doing a study, by including individuals who are at risk but don't carry the gene, it also prevents coercion of unwanted genetic testing, and there are ways in terms of randomization to be able to randomize actual gene carriers to an active intervention, and to those who don't carry the gene to a placebo group. And it's a little bit tricky, but it's been done in Alzheimer's disease and both familial and as you'll hear later on. So these are the two major. There's actually 3 studies going on that we'll be talking about later. So I'll just close. These are the questions that were posed for the symposium. Should high-risk status be disclosed to participants, and if so, what will the impact be? I don't think it's a question of should. I think it's really a question of what do the subjects or the participants really want, coming into the study. And I think there has to be a respect for this. And we see even to this day the uncertainty that exists amongst individuals who just know they're at risk, regardless of whether to undergo testing. And I see this as kind of a petri dish for genetics in the 21st century, as all these diseases will understand the genes, and everybody will be faced with this decision whether they have Huntington's disease gene or Alzheimer's or Parkinson's or none of those or just something else, breast cancer, whatever, to make this type of decision. And I think we have to be very cautious to do it. The boundaries between research and clinical care, that boundary moves, but the important part of the boundary between research and clinical care is the amount of risk involved. So when I see a Huntington's patient in the clinic, I'm making decisions and caring for that individual based on a lot of information and calculated risks over time. In a research setting, the risks are there, many of which have not really been calculated, and that's the importance of the research. So I think that would be my definition of the boundary, but just to say that boundary is going to change over time because of our knowledge. And I like this last one, but what's the best way to ensure that patients can provide truly informed consent? I like that because I hate the term informed consent because it assumes the patient's informed. And we don't know that. There's consent, and, we go through this process of supposing that they're informed. And actually, there's a lot of information being gleaned from this. So I would just say the best way to ensure is to take this process seriously, to do it, especially in a long-term study over time. We would reconsent our participants on a regular basis during the course of the study, and also try to learn a little bit about their understanding of the study, and done a lot of qualitative research from this. So I just want to close by thanking our major sponsors for this study. Human Genome Research Institute was our primary sponsor. NINDS was a big secondary sponsor, as was CHDI. During the course of the study, of course, thanks to the research participants. So there are 1,001 participants, about another 1,000 to 1,500 family members who came in. So there you're up to about 2,500. You had about 200 or 300 investigators, coordinators, consultants, people working in the laboratory. So there's almost 3,000 people, involved in this effort. And I would think same thing in the PREDICT study. So here's just a partial list of those individuals. And just a particular thanks to the leadership of the study. Actually, the study was conceived by my buddy Jack Penny, a neurologist at the University of Michigan, and he died after the study was launched a couple of years unexpectedly of a heart attack, and taken over by the study co-principal investigator Ann Young stepped in to take over. My colleagues at the University of Rochester, particularly Elise Case and Shirley Eberly, masterful, and David Oakes, masterful statisticians, my colleague Carl Kieberts, and then these were the real workers in the study who did part of this: Kevin Biglin, Steve Hirsch, Karen Martyr, Kim Quaid, Jane Paulson, and Carly Tanner. And just to close, there's another person that we lost over the period of time, and that was Elizabeth Thompson, who was at the National Human Genome Research Institute and actually ran the ELSI program. And I think a lot of what we are benefiting from ELSI was her ability, not just the legislation wasn't hers, but it was written in there, but to really kind of make it a reality and to be able to support research studies like Pharos and many others. And she developed Parkinson's disease and died. So thanks to all my colleagues in the Huntington Study Group. Some of you are in the room, and we'll see your pretty faces there. And I'll close, and do we make it in time?
So, discussion. Here are the microphones.
Thank you, Ida. I'm Professor White from CHDI. Thank you, Ida. I think it was a very interesting presentation. Of course, as you know, that you said, Pharos, which is a laboratory division for the field, is the observational study. And many observational study, as you said, has this paradigm of not disclosing the genetic status. And I agree with you. It's feasible, and it's indeed the common practice. But I would say that the current time is to discuss clinical trials, randomized clinical trials. And in that regard, I disagree with you. I think it's feasible, but it's not the best approach. So what I mean is feasible to keep the concealed status. I prefer the word concealed rather than blind, because the trials are blind and the two terms can become confused. I would say that this issue about this issue about that you mentioned about coercion and respect for autonomy can be highly difficult to understand, because people are not coerced to participate in a trial. If they are can make clear decisions. And these issues have been discussed. In fact, there is a paper published in Neurology by Kim et al., where all these topics have been discussed in the context of Alzheimer's disease trials that will be discussed. And ourselves, together with Bob Friedman from Colombian here, we have just submitted a paper on the same topic for Huntington's disease. We feel that there is no coercion, provided that people are fully informed. And I agree with you, informed consent is a very tricky method. But we are, and this is a very long discussion, we have to consider that we have interventions that are aggressive, that are not easy to administer. And this is where we are moving now. So it's not easy to do these trials in these situations of concealing, but it's not desirable as well.
Thanks for your comment. Other questions?
Other questions, comments?
Hi, Eric. Great talk. Quick focused question. Did you compare individuals who learned that they had a GAG greater than 37 versus those who learned that they didn't have that? Did you compare over time then how they did on measures of cognition, and was there a difference? And also those who, those who know they have the GAG lesion, repeat, and those who have it but don't know it. Did you look at the effect on cognition of that knowledge? Granted, there's bias to having learned it, but did you look at that?
Yeah, I don't think we have that information. You have to remember that we looked over this 10-year period, and most of this decision to be tested, although it was fairly uniform, happened towards the end of the study. So we didn't have a lot of follow-up after that period of time. But there's no question that the amount of cognitive impairment as we measured it was different in those that had the expanded repeat versus those who didn't have the expanded repeat. You're asking what happened after the testing. I don't think we would have seen much in the non-expanded group because they were operating optimally, at that period of time. But we didn't have those measures, and in the you're asking comparing that group. So we did this.
So the big is I know I have it. How do I do on cognition? I don't know I have it, but I do. How do I do it? How do those two groups compare?
Yeah. I don't think we have that information. My guess would be it would be hard. It would be small differences and hard to detect. Yeah.
A related question, also between those who have the repeats and decide to get tested versus not, were there any differences a priori? In other words, for those who decide to get testing, why some got tested and some didn't on their own?
In other words, how could we predict the decision to be testing amongst those who have the expanded repeats, like? Yeah. Well, I think that's also hard to say. Certainly affect and behavioral problems at the same time were there, but one of the big differences was the emergence of motor features. And there must have been a sense by the individual that something was happening, whether it was conscious or not. I'm just trying to give you an interpretation about what prompted that group to be tested. So a lot of this might have been a verification of their suspicions that they had, at that period of time. One thing that I've realized over now many decades in terms of caring for HD patients and people at risk is there is normally an amount of ambivalence that changes over time. And people will change their convictions over time from visit to visit, month to month, I'll bet you day to day. I have the gene, I don't have the gene. I'm going to make decisions based on that conviction, but that conviction changes. So I think it's a tricky thing to try to predict, but I suspect that in the context of the study, the emergence of motor symptoms and maybe cognitive abnormalities, if they could detect that, might have prompted the decision to actually undergo testing, and might have been more of a confirmation of what they expected rather than 50-50 yes or no chance, if that answers your question. Pardon? Well, we do have to look at the data. As I showed, the motor aspects do intensify in that population compared to those who decide not to be tested.
So, unfortunately, we just have time for one more question.
Okay, now let's go to Emily's one husband. I have a question about the suicidality and suicide ideation issue encountered. And my hypothesis is that kind of consistent with people who start experiencing symptoms, regardless of whether they're target status, because of higher suicidality and ideation. So it's probably nothing to do with study enrollment or participation. But do you think nonetheless that the study organizers, the PIs, the investigators, have some extra collaboration or responsibility to put people who they know that are having those thoughts, again, regardless of whether they know their target status, into counseling, or have some extra responsibility to culture people who are experiencing symptoms of depression, probably associated with their experience.
Well, the answer is for sure. And in fact, this was a preeminent concern that we had in beginning the study, during the course of the studying, and now that we see the data to this day, that although we are a little bit reassured that we could do a study like this, becoming complacent about this issue, and by the way, not just in Huntington's disease, but in clinical research in general, but particularly in Huntington's disease. Now, you have to remember that this is a pretty clean cohort. We excluded people in the beginning of the study. We didn't know their CAG repeat, but who came into the study who had any concern about suicidality. And that's what happens in clinical trials. You get rid of the people, or you exclude the people who are going to pose the greatest risk in the study. So we had a kind of clean cohort to begin with. Nonetheless, we encountered this in the study, both in those with and without the expanded repeat, but more so with those with the expanded repeat. And by the way, during the course of the study, if this ever arose, the clinical investigator would, we had provisions be incumbent to refer the patient or discuss this, but we usually try to do it outside of the course context of the study. So this was written into the protocol and into the consent document. And I'm not sure how often it was done at the sites. I mean, what we picked up, reported, I'm just kind of showing you the more gross changes, but the investigators spent a lot of time concerned about this and addressing this. Of course, most of these, and the nice thing about Huntington's disease researchers, they're also clinicians too, so they take care of these patients, so they understand these issues and they're attuned to the associated risk. So for sure, and although we feel reassured, we shouldn't be complacent about this, and also the notion that people feel really good after testing, that, and they adjust to it, and I do think they adjust, but I think that's another vulnerable period. Thanks.
Thank you. So now I'm really also very pleased to be able to introduce Dr. Jason Karlowicz, who comes to us from the University of Pennsylvania. And he is professor of medicine, medical ethics, and health policy and neurology there. His research focuses on ethical and policy issues in research and care of older adults with late-life cognitive disorders. He developed a new concept called wealth care, which is a novel model to promote cognitive health and maintain wealth. He also is a regular contributor to Forbes.com, in addition to his academic publications. And he's the author of a novel entitled Open Wound, The Tragic Obsession of Dr. William Beaumont. So thank you very much for coming.
thank you, Ruth. Thank you very much for that generous introduction. And thank you, Paul, as well, for having me here. It's a real pleasure. Oops, out here. So I know the program, the title is Using Human Models to Validate the Definition of Alzheimer's Disease, and this is my title. And actually, both titles are what I'm going to talk about. And I think, let me pick up on a remark that Irish Olson made during his talk at the beginning of his talk, which was there's a lot of stuff happening before we diagnose Huntington's disease. And if there's a takeaway point I would give you is that's the same story that we know in other neurodegenerative diseases like Alzheimer's and Parkinson's and Lewy body disease and FTLD, Pick's disease. And the goal of the fields, plural, is to figure out what it will take to find out that stuff and then intervene. And so someday, so I'm a clinician at the Penn Memory Center. I've co-directed. I see patients every Friday, more or less. And right now, to be diagnosed with Alzheimer's disease, I have to say that you're demented and that the cause of your dementia is Alzheimer's, or maybe it's Lewy body disease, et cetera. Someday, you will not have to be demented to have Alzheimer's disease. Someday, you will not have to be demented, impaired to have Huntington's. If we could go through all those diseases, instead, some test or tests will identify you with the disease. That's a key point that I think animates this conference. And the goal to achieve that is the route to that is the clinical trial. And I know normally we think of clinical trials as means to discover better ways to treat diseases. And in the studies that I will be talking about in my half hour, and ones that will be mentioned later by others, such as Diane, that is a goal. But the other goal of those studies is to use a word from philosophy, is epistemologic, which is to discover a new way to measure the disease and to change the definition of the disease. And the clinical trial is a very robust way to validate this new way to define these diseases. And if you want to, I'm an internist, geriatrician by training, and I saw this in my own practice when systolic hypertension in older adults went from being normal age-related changes to being systolic hypertension in elderly persons, a druggable target, courtesy of clinical trials that showed if you treat the blood pressure and lower it, you change outcomes. So you carve diseases out with drugs. That's what you're doing. And that's what's happening. That is what's happening in Colombia. The challenge in neurodegenerative diseases has been, and always shall be, that we had to first create it in mouse models because they don't normally get it, mice and rats. And then we discovered all kinds of wonderful treatments for mice and rats, none of which have worked in humans. And we've, while the labs keep on doing their work, and I like it, it's fascinating, the mouse labs, the field essentially arrived at we need to use a human lab. And it is individuals like the people I'm going to talk about in Colombia, as well as individuals in the United States who have the genetic mutation I'll be discussing, as well as individuals with Down syndrome who will become robust human models to try and validate this new way of diagnosing and treating these diseases. So that's using human models to validate the definition of Alzheimer's. That's that title. So now let me focus on this title, which is a discussion in particular of my title in the program, which is the Colombian Alzheimer's Prevention Initiative. And I'm going to be talking about the ethical, legal, and social issues of preclinical AD research in a developing country. And I italicized developing to emphasize that Colombia is a nation undergoing a variety of changes, and this trial, historically and socially, is very neatly embedded in those transformations that's occurring in Colombia. So this study is in place because for two reasons. There's a cohort of individuals, a population of individuals in the country of Colombia who sadly carry this dominant gene that puts them at risk by the ages of late 40s, 50s of developing Alzheimer's disease. But also, studying in that high-risk group is a great way to validate a concept of the disease and study it very quickly using an experiment, a clinical trial. A key message I'm going to give you over the course of my talk, which I'll say now and say it again, that I found when I reviewed this study is that the importance of building a community of engaged participants and the need for local engagement. People from the United States, funded by drug companies, et cetera, helicoptering in, landing in a developing nation and saying, we've got a great study, we want to do it. You can imagine the host of concerns that would raise. And one of the messages I'm going to show you is the amount of building that went on decades, years before this trial got started in order to assure that the study is responsive to the community's needs, which is the second point which you'll see me develop, which is developing a design of a study that fits with the current community standards, but recognizing that that study itself is going to change those community standards over time. And so I'll conclude with thinking to the future, where the future is really divided along what I think is probably a bright line. At the end of this clinical trial, the drug will either work or it won't work. And those are two very different futures that we're beginning to think about. So that's me. I'm at Penn. That's my Twitter feed if you want to garbage it up. And a little bit more about me. I don't have any IP or property holdings relevant to this. I do get support at Penn for our memory center to run clinical trials that are paid for by Lilly and Novartis, but not by Genentech or Roche, which is the sponsors of the study that I'll be telling you about. No paid consulting, and a variety of grant support, but in particular from NIA in collaboration with the Banner Alzheimer's Institute, which created the Alzheimer's Prevention Initiative, which is this group that is running this clinical trial. And the study that I am discussing that is occurring in Colombia is jointly supported by National Institute of Aging and Roche Genentech. So the Alzheimer's Prevention Initiative, run by the Banner Alzheimer's Institute in Arizona, has embarked on 2, soon-to-be 3 clinical trials whose goal is to recruit individuals who have an elevated lifetime risk of developing Alzheimer's disease dementia because of a gene that they have. And the second bullet is a study I won't talk about, but I'll start with, which is a study called Generations 1, which is enrolling individuals who have 2 copies of the ApoE4 gene, which puts them at heightened lifetime risk of developing late-onset Alzheimer's disease. So sometime in their late 60s, early 70s, they have a heightened risk. Estimates vary. We have a paper that just came out in PLOS that essentially says it's probably about 60-70% with a bit of a confidence interval around that. That's a high risk, but nonetheless, it's not the risk as high as those who have the autosomal dominant mutations, where the age of onset is younger, and if you have the mutation, essentially assures that at some point you will develop Alzheimer's disease dementia. By the way, that mutation is the mutation that still Alice Howland in the movie/book that won the Academy Award. When I think she was faculty here. I don't know why the book, she's at Harvard, but the movie they moved her to Columbia. I suppose New York makes for better optics, I'm not sure. But anyway, that was her mutation that she had. Okay, I'm going to be talking about the first study, which is a study that is testing a drug, crenuzumab, in individuals who have this gene. And they live in this country, the country of Colombia. In particular, they live in this one region of Colombia, Antioquia, which is in Colombia, it's in the Medellin region of Colombia. And it's a rural area in particular where many of these people live. And the story is, and they've traced back to the founder of this cohort, a Spanish immigrant to Colombia, a man, brought over the gene. And the rest is sort of history. Obviously, people didn't live typically beyond their 30s or 40s up until the 20th century, so the gene kind of lay hidden. But then people started living longer, and the gene began to make itself known. These are notes that might not be readable from the back, but they're more for me to help you understand the story of this gene. It's a fascinating story. It begins in the 1980s when a neurologist named Francisco Lopera, who's at the University of Antioquia, drives out into the country and starts to discover that there are people who have la bobera, which translates into the idiocy. And it's known in these villages and towns that people get the la bobera.
And around about this time, 1984, he's a resident. In his nights and weekends, he drives out there and he starts examining them and studying and learning about them. And essentially, kind of in classic bedside medicine, just starts to gather a host of cases and begins to see there's something going on here. And what is it? What is La Bobera? Well, what it is early onset Alzheimer's disease. It's what August Dieter had. She carried the presenilin mutation in Alois Alzheimer's first case report that gave us Alzheimer's disease. It's what still Alice has, et cetera, had, whatever. So by 1991, he has enough data to publish a case study and in the medical literature. And this is a great moment, which is researchers in the U.S. read it and they go, oh my gosh, this is a group at high risk with this gene. This is just the kind of group we want to study to try to better understand the genetics and pathophysiology of this, albeit unusual disease, which might help us better understand the more common disease seen after the age of 65 or 70. And with funding from a Fogarty Center grant, they begin to create a collaboration with the Colombian scientists at the University of Antioquia, Dr. Lopera and others. And I bring this up in this time here in this country because it's my understanding that the proposed budget for the NIH includes eliminating the Fogarty Center program. And I think it's very important to show how the Fogarty Center program, since the 90s, was instrumental in creating a community collaboration such that by the time drugs came along, like crenuzumab, that might change the course of disease, we'll find out in a few years, 5 years, there was a good groundwork set up for investigators to start collaborating and working together. In the country of Colombia, genetic testing is not the norm. There are no genetic counselors in Colombia. There are none. And for this particular disease, namely La Bobera, the standard in the community was not to know, was not to find out. Why? Concerns about employment, certainly, but really issues around family. In these communities, knowing that you had the disease essentially fated that you would not be able to have a family. And also, obviously, they're related to this as other fears of knowledge of the result, namely how people will treat you outside of issues of having a family. You can go on the web and type in 60 Minutes, the Alzheimer's Laboratory, and on the 27th of November, they broadcast a story about this study. And I think the title of that study says it all. That 60-minute show says it all. The Alzheimer's Laboratory. So these individuals are being used to try and study whether a drug can affect the natural history of Alzheimer's disease. There's a laboratory going on with them, but of course the hope is the drug may also therefore work and therefore benefit them. Colombia is a transforming country. A little bit about it. This region of Antioquia and the country of Colombia. Let me actually, I'm going to give you some statistics, but maybe a Huffington Post article is worth 1,000 stats. There was a Huffington Post article not too long ago, why you might want to retire to Medellin. Now, many of you might not think that. Medellin probably for many of you is associated with the drug cartel wars in the 70s and 80s. Those have ended. And Colombia and Medellin, which is the second most productive of the Colombia's states, has in the last 10, 20 years undergone an economic boom. The poverty rate since 2003 in both Colombia and Antioquia has declined from about 45% to about 20-25%. Similar trends are seen with unemployment and GDP. So Colombia is a developing nation. It's truly moving into the sort of picture of an economy similar to ours. So that'll come up at the end. So how do you do a study? Now, well, so here's the clinical trial. And I hope some of you say there's something wrong with this design. Most clinical trials are two arms. You get the drug or you get the placebo, and we follow you along. Sure, you might get different doses of the drug, but that's the way we design a trial. But key point, number one, shown in this slide is, given that standard of non-disclosure, no genetic testing, we don't want to know, how do you design a clinical trial where the entry criteria to get the drug is you have to have this gene? In fact, it would be unethical to give the drug to someone who doesn't have the gene. They would be expose themselves to the harms of intervention with no reasonable prospect that it would be a benefit to them. So you have two choices. You can get the gene tested and then tell someone you have the gene, you're eligible, or you don't have the gene, you're not eligible, but then you're up against that community standard that Dr. Lopera was well familiar with, and we were very confident going into the study that that was the community standard. And the solution is this, which is to create this third arm, which is those who don't have the gene who are automatically assigned a placebo. Everyone is concealed from their assignment. You don't know which arm you're in. And so effectively, enrollment in the study does not constitute disclosure of your genotype. That's what you've done here, okay? This design is being used in the Diane study, which in the United States, which also is enrolling individuals who have either presenilin and or amyloid precursor protein dominant mutations. And I know you'll hear later today from some people who are participants in the Diane studies. Of course, there's a challenge to this, which I'm going to show on the next slide in greater detail. But I just want to simply illustrate, there's a lot of text there, but the drug being tested is crenuzumab. It targets oligomers of amyloid, so therefore it plausibly is going right at one of the pathologies that we think is the cause of Alzheimer's disease, namely the amyloid plaques. Okay. Well, the challenge, of course, is that there are far more, even in the region where this gene exists because of its regionality and the relative rural isolation. The challenge is that if you just did an open call, come in and get tested, you're going to rapidly fill up that non-carrier arm, right? So what you start to get at the problem you can see is as people come in to be in your study, you say, well, you're not eligible. Why aren't you eligible? Oh, some results that I really can't tell you about, but, you know, because you've filled the non-carrier arm. You see the challenge there? You see that? So how do you titrate in the less common carriers and the non-carriers more common so that in a way exclusion, and this is, we struggled with this, and I talked to Pierre Theriault, the lead investigator, and Eric Ryman, and what we came up with was the creation of a registry. Namely, get word out to join a registry from which you will get genotype tested. You won't learn the result, only the registry knows it, and then the registry can selectively reach in and pull out some people without the gene and some with, and invite them in. And so you're sort of titrating them in to the study site. You follow that? That's key. And so there's this infrastructure issue. So these are the many things that were done to get the registry going in the country of Colombia, building on Lopera's existing cohort study. And this is just one of the ads that ran on bus kiosks and outside, which is reproduced here. So in Spanish, obviously, familiar Alzheimer's manifests in adults, young adults, and the risk with heredity is very high. If you have a family member with progressive loss of memory before the age of 60, communicate with us, call 219-6664, et cetera. And so people were calling in and saying, yeah, you know, so they would join the study, join the registry. And so this is the registry that was created. This is roughly as of just a few months ago. And so it's out of this group, obviously, that you're going to, let me see if I can get this. Whoops, at least get the, you know, so you're going to sample some of these and some of these to bring them in then to come in to be screened, et cetera, and really keep track of how you're filling so that you don't rapidly fill that non-carrier arm. And this is what you get. So they set up 3 satellite sites to administer the study, enrolling 2 to 1 people who are carriers to non-carriers. And this is just as you would expect. They're not, one of the criteria to be in is you have to be cognitively normal, hence the age of people is basically 40 years of age. Age of onset is roughly late 40s or so. So what they're trying to do is capture people, enroll people who are not yet impaired with the goal of seeing if the drug delays the time before people develop cognitive impairment. And the primary endpoint is cognition, not conversion to dementia, which is a very interesting issue from a policy perspective we can talk about. So the study's ongoing. It is up. It is actually filled now. There are all arms are filled. People are getting drug or placebo. Testing is going on, and the study will end roughly in 2022. So in 2022, if the study works, we will have validated that targeting amyloid in individuals at high genetic risk who have this disease, as Ira said in the beginning, happening before they have the disease, as we know it clinically, if the drug works, we will slow the natural history of decline. And we'll talk later at the end. I'll bring up the implications certainly are substantial for individuals with this gene, but they're substantial for everyone in this room because garden variety Alzheimer's disease also we think is mediated by amyloid pathology. So this is a great human model for the more general problem of Alzheimer's disease late onset. So I have here the rates of observed versus predicted pre-screen fail rates, just kind of before you come in and get detailed screening, were higher than expected because of especially prohibited medical conditions of high rates of thyroid, hypothyroidism, et cetera. Already incipient cognitive impairment, namely mild cognitive impairment or MCI, and individuals who, despite efforts, which I'll talk about later, to tailor the informed consent process to fit their educational level, many people here having at most kind of a grade school education, simply couldn't give informed consent. They couldn't work through the details of the study, even though they were not cognitively impaired. Screen fails, I've already mentioned, once you start testing people for other medical conditions. But I think the very interesting finding is this one here, which is the investigators predicted, excuse me, there's a small fly, and or I'm having a delusion or something, or illusion flying around my head. The investigators had predicted a 25% dropout rate from the study. It's a 5-year study. It's a lot of work, and these people, many of them, have to travel a fair amount, even if they can get to one of the satellite clinics in Yaromal or Armenia. And yet, the dropout rate so far is less than 5%. So, a very motivated group of people wanting to come in and come back. So, let me go through some of the ethical issues: minimizing risks, respecting autonomy, and justice. And then I'll wrap up talking about what if the drug works or what if the drug doesn't work. That clock is an hour slow, right? So, okay. So I've talked about the 3-arm design, no more details on that. I've talked about how a registry then becomes key to do this. Respecting autonomy. This is a quote from the Fogarty Health Matters. This is the reference. If you can't read it, just go and type in Global Health Matters Fogarty. Look at volume 13, issue 1. It's a great article about the Fogarty work in Colombia. And here's Dr. Lopera speaking. Our patients in Antioquia have participated in various studies for almost 3 decades. They are family to us. They have seen many relatives die of the disease, and they understand that it is incurable. They're very hopeful about this drug trial. When we also have hopes, but it may do absolutely nothing. Yet even in the worst case scenario, this information will be important for the future. He really has developed the statement, they are family to us, is a very legitimate statement on his part and his team. They really invested since the 80s a relationship with this community, as opposed to helicoptering in with a drug company and running a study. No offense to the drug company, it's just that concern exists that we, the United States, pop in, as particularly we tended to do for a while in South and Central America, and do what we want and then run away. The documents had to be translated into Colombian, Spanish, and won't develop this in much greater detail, but they created a picture guide in addition to that wonderfully, typically unreadable informed consent that actually was, and we've been using this in other studies in Alzheimer's prevention space, namely, there's the consent form, but here's the guide that you really need to read that tries to explain everything in a clearer, more reader-friendly way around issues of either the gene and or, in the case of the A4 study, biomarker testing. But here I think are the points I want to wrap up on, which are issues sort of related to justice, which is treating equal people equally. So Colombia does have a universal healthcare system. The challenge of that universal healthcare system is it's very much divided along public-private. Everyone has access to public health care, but there's a robust private system. And so you can go, for example, to Bogota and find the private hospital, which is a little like something in the Upper East or West Side of New York. Or you can find the public hospital, which is a little like something maybe on the downtown of New York on the West Side. My point being that extremes of wealth and poverty have created, while everyone has access, the quality of care they get. And while that universal access has brought health care that did not exist to the rural regions of Colombia, it still is a challenge to deliver quality care. And so now that people are in this clinical trial, the investigators felt an obligation that for the duration of the trial, particularly as adverse events occur, et cetera, that they needed to provide health care. So where the system of health care that exists couldn't provide health care, the sponsor committed to making sure people got the health care they need for the duration of the trial. They also implemented a social plan that will be in place post-trial. That's what it's called. Essentially, this is the psychological and social care that has been shown to clearly benefit individuals with dementia. And the plan is that this will remain in place post-trial. Obviously, it's needed during trial as people start to get impaired, because some will. They will. They will even develop dementia. But you need it in place afterwards, because think about what you've done. By walking in and now creating this trial and testing, et cetera, regardless of the outcome, you've raised awareness. You've identified a group of people who are now tuned and sensitive to the issue of my cognition. And so no matter what the result of the trial is, they're in a new space of learning to live with what is a failing brain and how to live with it. And then this third bullet is the sustainability plan, which is access to the therapy if it's effective. And the company has made a commitment to work with the Colombian authorities to assure that the drug can be made available in terms of its pricing and access should this trial be successful. The second challenge that the company and, frankly, myself are beginning to work on and others is to go from having no genetic testing to having genetic testing. In a country where there was no genetic testing and the standard was we don't do it. If this trial works, in particular, genetic testing will become the norm. Colombia is an aging nation. These graphs just simply show it. They're a lot like we were around 1960 or 70 in terms of the demographics. And so I wanted to kind of think about the broader implications of this trial in the country in my last few minutes here. If the drug isn't effective, crenezumab does not work. It does not change the natural history of the disease. Well, still awareness will be raised. I showed you that bus kiosk, for example, advertisement. So I think what will start to happen is La Bobera will transform into Alzheimer's familiar, to progressive memory loss, or even just Alzheimer's. In other words, Colombia, this trial, this work, will transform how Colombia thinks about Alzheimer's disease. And I think they'll kind of become like we were in the '84. That Alzheimer's will be something that you have to have dementia to get. Why would you want to know about if you have it, et cetera? And I think Colombia will sort of rapidly enter where we were in the 80s. If the drug is effective, not only will Alzheimer's, La Bobera become Alzheimer's familiar or Alzheimer's, there will be this thing called preclinical Alzheimer's. Namely, if you get the gene tested and you learn you have the gene, even if you're asymptomatic, we can now intervene with this drug and change the natural history of the disease. But that will be a small population of people. But for the larger population, this trial now says if you have amyloid in your brain, maybe this drug will work to prevent you from developing Alzheimer's later in life. And so what Colombia will struggle with is a broader dissemination of the Alzheimer's concept from being just having dementia to having a gene and or this amyloid marker which says that you someday could develop Alzheimer's disease, dementia, and therefore you need this drug. So Colombia is on the edge of, by 2022, a real transformation in what they think about when they think about Alzheimer's disease and how they live with it. In the setting, of course, of an aging nation, much like ours, where there will be more and more patients needing more and more help. I want to thank the Alzheimer's Prevention Initiative team, particularly Pierre Theriault, who led this trial, Francisco Lopera, who I've mentioned, and Eric Ryman, Pierre's colleague, and also my good friend and colleague at Banner, Jessica Langbaum, who's been instrumental in this and other studies. I have a few minutes for questions.
Oh, yeah.
Great. Okay. So thank you very much.
This is Jason. That was really good talk.
Well, you're welcome.
I have two questions. One is, how do you think the unexpected high rate of exclusion of people into the trial relates to the unexpectedly high rate of maintenance or low dropout rate? Do you think the two may be related to each other?
Yeah, that you've selected for the healthier group of people who, therefore, can stick around for the study. Yeah, I think that those two certainly probably have a causal relationship. Yeah.
And then you started to talk and ended your talk with this concept that Alzheimer's disease will no longer be restricted, that term will be no longer restricted to people who have dementia.
Right.
That there will be that definition based on your genetic status or your amyloid status. Is that a concept that has been talked about or reviewed with the community of people who may have amyloid or may have genetic markers for disease? I'm asking because I'm not sure that people want to be labeled as having a disease prior to ever having any symptoms. I'm just wondering about that.
It's a great question. So in the States, of course, we think that there is this that someday we will discover and validate this thing called preclinical Alzheimer's. And that's what we say as researchers. I'm one of them. I think that the cultural experiment that's going on both in the United States and in Colombia is, will our culture embrace that the Alzheimer's label can be used on someone who's essentially normal? So, for example, someone in this, genes are tough because you have the genes from the moment you're born or you don't. But let's talk about a biomarker. If I learn I have a head full of amyloid and I'm in the A4 study, do I have do I have Alzheimer's disease and I'm just not yet symptomatic? And many people in the science field would say you do. But I don't know how our culture if our culture will take that term and allow it to be used, or will we say, well, I'm at risk for Alzheimer's? And we're in for a big language experiment, not just in Colombia, but here. And that's one of the things I've been keenly interested in studying, how that term will spread. Yes, spread. Yeah.
So I wanted to thank you for that presentation, because it was really a wonderful display of just how complicated conducting this kind of clinical trial can be, especially in foreign context. And you spoke to some of the cultural context in the community where this is much, much more prevalent. And I just wondered, following on the previous question, if you have collaborated with any anthropologists or folks who work locally to better understand how the community affected by this gene might interpret specifically genetic knowledge, if this is not commonplace in the current context.
Yeah.
And also in relationship to that, whether there would be adoption of this kind of identity that you just described, and if you could just go into a little more detail about the belief systems and understandings that might be at play there.
I want to. The answer is, in Colombia, no, we have not. I have not been able to set up a collaboration with an anthropologist or similarly trained social scientist to do just what you've said. I want to. If you're one, we should talk, or if you know someone, we should talk. In the United States, we have 2 studies going on in collaboration with my dear friend and colleague, Pamela Sankar, who's an anthropologist. Those studies are called Socrates, the study of knowledge and reactions to amyloid and or ApoE testing. And we're doing just that. We're trying to understand the participants' perspective on the meaning they make of this information, and who they share it with and how it affects their relationships, both people who learn amyloid and or gene results. This is, I will leave it, there is much more work to be done in this space, and I would really love to see the field of medical anthropology and others walk into this area. It needs a lot of work. There's something special about the brain, and when it fails, it's very different than the rest of our body, so, in terms of identity, et cetera.
I imagine in the course of the trial that's 5 years long, that there's a risk of disease onset.
Oh, absolutely, yeah. And especially
so how is it treating the protocol, but more importantly, from my perspective, how is it treated in informed consent?
Well, in the informed consent states that over time you can develop cognitive impairment, you will be tested, and if people do convert or otherwise show clinical change, then that is a clinical event that needs an assessment and workup, and if it is indeed Alzheimer's, et cetera, then steps appropriate for their care. I mean, and I don't want to belabor that, but the cognitive impairment may be from some other reason other than Alzheimer's. The odds are it is, but so they need a clinical assessment. Yeah, that's part of the protocol. Sure. Yeah. Ira?
Thank you.
Yeah.
Just, you mentioned there are no genetic counselors in Colombia. I'm not surprised, although there's probably a few clinical geneticists and physicians and things like that. But just a note about our lack of preparedness for venturing into this whole area. In the United States, there are 2,500 genetic counselors, most of them trained at Cyril Lawrence University.
So they're all here.
Well, I'm just saying there's a lot of, this is kind of the epicenter. 1,200